So my name is Anton Bushuiev, and as it was mentioned, I'm a first-year PhD student, supervised by Josef Shivets at the Czech Institute of Informatics, Robotics, and Cybernetics in Prague. And today I'm going to present the results of our collaboration with the Loschmidt laboratories, which are at Masaryk University in Brno, and also the Institute of Organic Chemistry and Biochemistry in Prague. And as you can see, my talk is going to be about designing protein-protein interactions. So let's start with what they are. And please, since it's a multidisciplinary talk, it may be hard probably to understand it during the talk. So feel free to ask questions if it's not clear. And so to start with protein-protein interactions, this beautiful animation shows how coronavirus attacks or penetrates a human cell. So in the first place, we can see that coronavirus has these spikes, which are visualized here in orange. And each of the spikes indeed consists of 3 proteins kind of sticked together. And we can see that coronavirus uses these spikes to attach to proteins which are visualized in purple. And these proteins are receptors on the surface of a human cell. And then we can also see another kind of proteins here in blue. These ones, and they are called antibodies. And as we can see, they are produced by the human immune system to block the spikes so that coronavirus cannot use its proteins to attach to human receptors and get inside. And this video doesn't show some, let's say, special example of a system which is abundant with protein-protein interactions, but vice versa, these interactions between proteins, or more widely known as protein-protein interactions, in fact underlie the treatment and the development of many diseases such as stroke, cancer, or Alzheimer's. And our collaborators, in terms of the Stroke Brno Consortium, designed drugs against stroke. And as we can see here on the right, stroke is essentially a medical condition when a blood vessel is blocked due to the cluster of these proteins which are called fibrins. So our goal is to degrade this clot or to break it down and to restore normal blood flow. And so what we can do is to use the staphylokinase protein. So staphylokinase is a thrombolytic drug candidate. And what we can do is to take some concentration of the staphylokinase proteins, put them into the bloodstream, and then in blood, staphylokinase proteins start interacting with another proteins, which are called plasmins in purple here. And then together, these 2 proteins form what is called a complex. So further, these 2 proteins operate together as a single unit. And as we can see, they together catalyze or stimulate the production of more of plasmin molecules and on the surface of this clot. And these plasmins essentially eat this cluster of fibrins and help to restore the normal blood circulation. And this mechanism is very well studied, and but for many years, and the staphylokinase is known to be much cheaper and also safer than state-of-the-art thrombolytics. But it has an issue, and the issue is that staphylokinase doesn't really want to interact with plasmin as much as we want. And it's exactly our goal to make staphylokinase somehow more attracted to plasmin, to achieve more effective thrombolysis. And before I'm going to proceed for our approach to this problem, let's firstly discuss what actually proteins are. So what are all these objects here? So let's take a closer look at this staphylokinase protein. So now we can see it again here, but in a more detailed view. So first of all, we can see that proteins are in fact large molecules or macromolecules. So we can see that there are a lot of atoms connected with chemical bonds. And it's not just some random cloud or bunch of atoms connected together, but in fact, it's a sequence of particular building blocks, which are called amino acids, visualized here on the left. So we can see one of such building blocks here in green. And this is amino acid, which is called phenylalanine. And we can see that it has this aromatic ring. And so protein is in fact a sequence of such building blocks connected together and then folded somehow or twisted in 3D. And since it's a sequence, it's also often visualized like this to show common folding patterns, which are visualized here with these arrows or these spirals. And now let's get back to our initial question of how to make staphylokinase more attracted to plasmin. So here on the left again, we have a staphylokinase protein, which is a thrombolytic drug candidate. And here it interacts with plasmin, which is a protein present in blood. And if we take a closer look at this, what is called an interaction interface, where proteins touch each other or connect, we can see that this interaction is dependent on the properties of amino acid building blocks. So that's why we can approach our question in the following way. So we can try to achieve higher attractiveness of staphylokinase by modifying these building blocks or substituting them. Technically speaking, these substitutions are called mutations. So for example, we may focus on these 3 amino acids, and we can imagine that we, for example, may mutate this amino acid with some smaller one, or to replace this amino acid with some other one which has, let's say, an opposite charge, and it may contribute to this attractiveness between 2 proteins. But of course, as we can see here, this introduces a combinatorial problem. So even if we only focus on these 3 positions or building blocks, it's already 20 to the power of 3 of possible mutants or combinations of different building blocks. And so, of course, even if we consider further the whole interacting interface, it leads 20 to the power of n combinations, where n may be large. And moreover, this whole system is highly flexible and dynamic, so it's really hard to somehow precisely calculate or predict the effects of such mutations. So what would be the attractiveness of proteins if we substitute some of these amino acids? And so now a bit more technically, so that I do not keep saying attractiveness. So how can we quantify how well proteins interact? So it's typically measured by delta G, which is binding energy of proteins. So here G stands for free energy, and delta is the difference between the energy of the system when 2 proteins are apart and when 2 proteins are together interacting. So the lower this energy is, it means that the more stable the complex or this interaction between 2 proteins. So we want to minimize this quantity. And so now it leads us to finally to the definition of our problem a bit more technically. So what we want to predict is delta G, which I will explain in a second. So essentially, we are interested in developing or using a machine learning model, or let's say a neural network, which can take the following input. So some 3D structure of 2 proteins interacting together, or protein-protein interaction. And then we also want to tell our neural network that let's say we are interested in a mutation of amino acid 34 from this protein in blue. And we know that the native building block or amino acid there is serine. And we, for example, interested what would happen, or what would be the binding energy if we mutated to glycine. So now we have these 2 kinds of input, and we want to predict a single number, which is delta G. And it's just delta G, so binding energy, but we add the second delta for the difference between the native structure and the mutated one. So again, lower delta G is, the better for us, because proteins interact better. And the delta G measures the difference or how much we improve or lose upon introducing a mutation. So it's a subtraction between the mutated binding energy and the original one. And this delta G values vary roughly in the range between -12 to 12. And for example, we can see that the model may predict this slightly negative value for the mutations that we have on input. And since it's slightly negative, it slightly improves the interaction or makes 2 proteins more attracted. And so we consider it, let's say, a slightly good candidate. And what is very important to mention is that the distribution of delta G values typically looks like this. So the majority of mutations, of random mutations, are in fact not interesting for us because they only disrupt the interaction and they have positive delta G. But some of them really improving. This is the binding, and we are interested in identifying or detecting them using a machine learning model. And so now what is very good for us that there exist such data sets which exactly have the data we want. So they contain some protein-protein interactions, let's say 3 proteins interacting together. And then we know that if we mutate amino acid 21 in some of the proteins denoted here it's at C, let's say this is this one. And if we mutate amino acid 21 from cysteine to valine, then delta delta G is slightly negative. So it means that it slightly improves the interaction. Or another example may look like this, and it has a positive delta delta G, so it's not really interesting for us. And then, as we can see, such data is very scarce. So experimentally, it's really hard, time-consuming and expensive to obtain such data, to measure them in a lab. That's why such data sets contain only several hundreds of interactions and only thousands of mutations associated with them. And so finally, our approach is to break down this limitation of scarce data by learning from a lot of unannotated data. That's why in the first step, we collected this large data set of protein-protein interactions, which we call PPIRef. And as we can see, it contains 2 orders of magnitude more interactions. And as I will show in a second, from these thousands of interactions, we can really construct millions of training examples. And so how we obtain this data is that we basically mined all the protein structures that humanity publicly knows from the database, which is called Protein Data Bank. And now, before I'm going to continue with how to really leverage this large unannotated data for machine learning, it's important to mention a certain aspect of its preprocessing. So working with such large data, it's really important to ensure that we evaluate generalization and not just how well model overfits to training data. And for example, if we take a look at the data set, so here are 2 entries that we have. And visually, we can see that they represent identical interaction, but these are different entries in the data set. So we need to somehow separate them. So to have both of them either in the training set or the test set. And it turns out that it's not really trivial. And so, for example, if we compare just sequences of these proteins, for example, if we look at this pink protein here and the pink protein there, and they really have different sequences. So amino acids or these building blocks are very different. If we align 2 sequences and then compare the matches at corresponding positions, we only have 26% of identity. So it's hard to recognize programmatically that these interactions are the same just only by comparing sequences. And another intuitive approach may be to somehow align the structures globally and say that if they have high alignment scores, that they are similar, and if they have low scores, then they are not. But this approach also doesn't work because it doesn't scale to large data. So if you want to really compare thousands of interactions, we need something faster. That's why we developed a method which we call IDs. Essentially, it converts interactions to vector representation and compares distance between them. So here we can see that it calculates low distance for these 2 interactions, which means that it can detect that they are really near duplicated. And our approach is roughly 500 times faster than alignment-based methods, and it also detects and it also has quite high precision and recall compared to standard alignment-based approaches. So now we have a large data set of unlabeled protein-protein interactions and also a tool to preprocess them, so to make splits and also to remove biases by removing near duplicates in data. And now we can finally proceed to machine learning. And so now the question how to really leverage this. So what we know from the traditional application domains of machine learning, such as natural language or computer vision, that masked modeling works very well. So some similar approaches stand, for example, behind the development of ChatGPT, was a successful models. And so essentially working with large text data, what we can do is to create this kind of artificial training examples just by taking sentences without any annotations and then masking some random words and training the model to predict which words are missing, like for example here. And so now we have the question, can we adapt this to our problem, so to the problem of protein-protein interactions? And I want to show that the answer is yes, and I believe that it's probably this kind of masked modeling is probably even more suitable here for proteins and mutations than it may be in other domains. So here is the illustration of the self-supervised pre-training from unlabeled protein-protein interactions. So in the first step, we sample some interaction from our large data set, and then we store, and so our labels are the types of amino acids here. So we store a vector C, and the length of this vector is equal to the number of amino acids here in our interaction. And then the entries are just, let's say, from 1 to 20, which correspond to different standard amino acids or those building blocks. And then we convert this interaction to a graph-based representation. Here, each node corresponds to one amino acid, and then we connect it to a predefined number of nearest neighbors. And then each of the nodes also has orientation, and this orientation intuitively captures where amino acids look. So, for example, before I showed, I discussed the example of amino acid which has this aromatic ring. So here it captures where this ring is oriented. And then, of course, each of these points is associated with 3D coordinates. And then what is important is that we mask some of these amino acids randomly during pre-training. And so then we obtain this masked version of our labels. So the vector where we just replace some of the types with zeros. And then we apply quite large neural network, which is based on the successful and well-known transformer architecture, but it's also adapted to work with these three-dimensional graphs. And then after that, we obtain embeddings or hidden representations for each of these amino acids. And then what we do is that we take amino acids which are masked and just apply a simple linear classifier to predict the probabilities of amino acids. So here we have the types of amino acids hidden for the model. So model doesn't know what are the kinds of amino acids here. And then it predicts this two vectors corresponding to these two positions. And each of these vectors is of the length 20, which corresponds to this categorical probability distribution over 20 possible amino acid kinds. And then what we want to do is to, during training, is to maximize the likelihood of the native amino acids. So here, one of these probabilities corresponds to the amino acid, which is indeed here in the structure, and the same for the second vector. And so we apply negative log likelihood loss just to, so that model learns which amino acids are missing. So intuitively, the loss is 0 when the model predicts 100% probability for the true amino acid and 0 for the rest. And so to summarize, we have this approach how to sample really millions of masked examples and to train the model to reconstruct amino acids which are missing. Here, so this is a very powerful approach in natural language or computer vision to obtain very strong numerical representations of data. But here I believe that it's even more probably suitable because we can quite easily adapt this approach to predict the effects of mutations. So now again, if we get back to our delta G, so delta G is quantifies the effects of mutations, whether they improve binding affinity or not. So now on the input, we again have some 3D structure which describes the native interaction, but now we also have a mutant. So it stores the types of amino acids, but in the mutated structure. So it compared to this to original representations, some amino acids in this differ. And what is important that now we do not mutate some random amino acids in the structure as it was done during pre-training, but we masked exactly the positions which we are interested in, so the ones which are mutated. And then again, we predict the probabilities. And intuitively speaking, now we can reason that if the model predicted higher probability for the mutated amino acids, then they are more likely, and it means that they may be better for the interaction. So the binding the energy may be lower or better. Technically, what we can do is that we can estimate delta G as this difference between the estimate of the joint probability of the native structure. So we just sum the probabilities corresponding to the native amino acids. And then we also do the same, but for the mutated ones. So it would be two different numbers here. And then we can see that if the joint probability of the mutated structure is higher, then we will get a negative number, which corresponds to a negative delta delta G. And it means that the mutation is good for us, that it improves the interaction. And then we can regress through this difference to fine-tune it on our small annotated data sets of mutations, but also leveraging pre-training for millions of examples. And so now let's take a look at the results we obtained. So first, we evaluate our approach on the standard dataset of mutations on protein-protein interactions. It contains roughly 300 interactions and 7,000 of mutations. And we compare different methods in the three categories of metrics. So first, we estimate how well the predictions of delta delta G correlate with the true delta delta G values. So we want the model to basically sort the mutations well. Then another category of metrics is that we measure how well the model can recognize good mutations, or the ones that have negative delta delta G, because it's practically important to identify them. And then another metrics are just how well the delta delta G values are predicted in terms of the absolute numbers. And then we compare our method against machine learning baselines. Some of them only require sequence on the input, and some of them are structure-based, so they're really diverse. And then we also have a simulation-based approach. So it's 5 orders of magnitude slower than machine learning approaches, but it sets kind of an upper limit on performance. And as we can see, this simulation performs the best. And among machine learning approaches, our method achieves the state of the art. And then we also confirm these results by assessing the performance on 2 independent case studies. So the first one is already well known to us. So here again, we have this staphylokinase protein, which is a thrombolytic drug candidate. And here it's interacting with plasmin from blood. And here in this system, we have, so if we introduce mutations here to staphylokinase, we know some of the effects which were reported before in literature. And we know the effects of 80 mutations. So 6 of them, let's say, are very good. So they enhance thrombolytic activity at least twice. And 14 of them just improve the activity, and the rest are just bad, not interesting for us. And what we do is that we make predictions of delta delta G with our model for all these mutations, sort them according to the predicted values, and then just look at the ranks. So we want to have good ranks or low ranks for the mutations which are known to be good. And what we can see is that our method predicts 2 mutations which increase activity at least twice as the top 2 hits. So in this pool of 80 mutations, the ones which are known to be very good are at first 2 positions. And then we also make a similar evaluation, but on a different case study. So here we have a human antibody on the left, and the spike of coronavirus that I showed before. And in this example, we have 500 mutations, and we know only the effects of 5 of them. And here in this table in bold, we have the values which are below 10%, so which are in the top 10% of predictions. And we can see that our method identifies 2 of them and tends to score mutations which are good with lower ranks. And also in this pool of 500 mutations, the one so the best hit by the model is indeed a favorable mutation. And so further, we created a web interface for our method so that the researchers can freely try our method on their data. So here we have the input that I just showed before. So on the input, we need some 3D structure of a protein-protein interaction. and then we specify some mutations to the structure, and the web predicts delta delta G values along with some visualization. And on this, I want to end my talk and proceed to discussion. But before that, I want to thank you for attention, and also mention that we all are keeping our fingers crossed now for the ongoing wet lab validations of our joint designs with Loschmidt laboratories. So here we can see a photo from Brno, and in these boxes we have this modified or designed versions of the staphylokinase protein, and they are currently ongoing wet lab validation. Thank you very much.
So thank you very much, Anton, for the talk. I'm going to now switch off the recording for the discussion. So stop recording.